Now I'm going to talk about what determines the protein structure. And so here we have a normal protein, and this normal protein can be denatured. And when we talk about denaturation, do you see this normal shape? The denaturation is when the protein loses this normal shape, and the renaturation is when it comes back to the normal shape. So there are many ways to denaturate a protein. So for example, changes in pH, in the concentration of a salt, or the temperature. One of the reasons why we cook our food is because when we warm up the water, what we are trying to do is to take the microproteins and denaturate it so they cannot able to renaturate again, and they are not toxic for us anymore. So this is why we cook our food. And not only we can do that, we can also destroy the normal proteins, the normal shape, using pH and using different salt concentration. Something that I want that you remember is that in addition to the primary structure, there are physical and chemical conditions that can affect the protein structure. And I give you a few examples. So changes in the environment, like pH, temperature, etc., can affect the normal shape of the protein, and you can denaturate it or renaturate it. And the loss of protein native structure is called denaturation, and this will cause a biologically inactivate the protein. Now I'm going to talk about protein folding in the cells. This protein called chaperonin is very important. It's very difficult to predict the final shape of a protein only from the sequence of amino acids. So living organisms have embedded this protein. Let's say this is an assembled machine that allow proteins to properly fold. So they have a cap and they have this hollow cylinder. When you remove this cap, a protein going inside, and here in the hollow cylinder, this protein is going to have a lot of chemical interactions, and all these chemical interaction will allow them to properly fold and to go outside properly fold. And this is something very important because this disease like Alzheimer, mad cows disease, and other ones are related to an improper folding of the protein, so a misfolding can create diseases. So I want that you remember that first, it is hard to predict a protein structure from its primary structure. So to solve that problem, living organisms have created an assembled machine that are called chaperonin, that are protein molecules that assist the proper folding of other proteins. And diseases such as Alzheimer, Parkinson, and mad cow disease are associated with misfolded proteins. In this slide, I want to show you the different steps of the chaperonin action. And in the beginning, here you have an unfolded protein going inside of the hollow cylinder, and at the end, this unfolded polypeptide is going to come outside correctly folded. But let me show you in a little bit of detail what is happening. So in the beginning, you remove the cap, as I said before, and the unfolded polypeptide enter to the cylinder from this end. Then the cap attaches again, causing the cylinder to change in shape. This hydrophilic environment creates the right environment for the folding of the polypeptide, and at the end the cap will come off and the properly folded protein is released.